So I'm showing this, let me ask you a question. If you're familiar with the AOP concept, adverse outcome pathway, raise your hands, please. If you're familiar with it. So there's a good amount of people here familiar with it, but there's also quite a few people not. If I can, okay, there's people who are not really sure what an AOP is about. Please raise your hands. Be honest, it's okay. All right, fine. So adverse outcome pathway is pretty much getting a consensus, including the regulators, importantly, of the way we should go about doing risk assessment of chemicals, and that probably is going to apply to nanosafety as well. And I want to emphasize, it's a knowledge-based framework. Now you probably have a question, what does that mean? What is a knowledge-based framework? We don't have a picture of one of the pathways? We do?
At the end.
At the end? Oh, okay. Well, we'll come back to that. So the idea is we have events happen in the world. We have initiating events. And the initiating event might be a drug molecule coming along and interacting with a protein that it was designed for, and it has an effect. And that's why you're taking the drug. That's a key initiating event. But it also might happen that a chemical or a nanoparticle may come along and have a key initiating event after the exposure in the human body, and that triggers a pathway. So in nanosafety, like what Vladimir is working on, it might be the nanoparticle forms this protein corona complex, and then that has that interaction with the membrane that the modeling is trying to understand, because we really need to understand the key initiating event, and then that nanoparticle is in the cell, and we have to study what happens there. So in the adverse outcome pathway concept, we're going from molecular key initiating event all the way through to the adverse outcome, which might be, for example, cancer in the whole population and what's happening in the environment, right? So it's many scales of time and complexity. It's a very complex problem, right? So we're dealing with a complex problem. It's more complex than predicting the weather. It's a lot more complex than sequencing the genome. So it's a knowledge problem. So we need a knowledge framework. This won't be a database solution. It won't be a modeling solution. It'll be putting the different components together, like we'll do today with the tools. The tools are important. And then having discussions around the evidence, and I really encourage you to do that, to come up with taking a scientific-based judgment on the evidence we have to do a risk assessment. And the AOP knowledge-based approach tries to, based on evidence described from that key initiating event through intermediate events, through end events, what's the actual model we have of the world and how that happens. So I'll give you an example of an intermediate event. So a lot of times safety is not necessarily about that key initiating event. We deal with all sorts of perturbations on our system all the time, and we're very good at dealing with that. But as we age, our stress response and recovery changes, right? So actually the reason I might have an adverse effect is not necessarily the key initiating event. It's how does my stress response pathway at that dose respond? And I might have been dealing with parties after conferences for 20 years, but in the future with personalized medicine, and we will have personalized toxicology. With the big data analysis, we're going to have my intelligent agent advising me, Barry, you're a little older now, and that stress response pathway is just not good enough right now. So you lower your dose and go home, right? Earlier, right? So that'll be based on the AOP. We'll do risk assessment, but we'll also do personalized medicine on it. That's what we're talking about. So when you use these tools today, think of them as components of that. We'll have data around an effect. We'll have we'll have a kinetics model that tries to then look at the dose response around that particular effect with that particular reference compound. That's the future where we're going. Now, it's not all in place. So, for example, this AOP area is incomplete. We don't know actually about all the genes involved in toxicology. We don't know what all the AOPs are. We don't know what all the effects are. And what we certainly don't know is we don't have it quantitative. So one of the big moves with the kinds of tools you're using here today is to make this quantitative. To make it quantitative, we're going to have to be able to deal with uncertainty. Because to do a risk assessment, it's not just about effects and exposure, but all the uncertainty and the factors and the key events leading to adverse outcome we have to understand. So I just want to take, before I hand over to Vladimir, 5 minutes to try and explain that. So this is a picture of my cat Pixel, right? And this we go for a walk because I might mention this and people might think cats don't go for walks. Well, actually, she does go for a walk when you can find her, and also with her sister. She has a sister actually. When that's a more challenging part to find the sister or to keep the sister with her. But she's a pretty brave cat. So we go for a walk, and you can see there she's pretty happy about it, right? So I ask a question to you. Just think about it for a little bit. Maybe write it down on your piece of paper. When we go for, if you believe me now that we actually do go for a walk, which might have been your first disbelief, how likely do you think, what is the most likely distance from the house that we reach on our walk? And I and my 2 cats go for this walk. Just write down the answer and let's see what your answers are. I'll give you a few seconds. What's the most likely distance that myself and the 2 cats reach from our house when we go for a walk? Don't think about it too long, just write it down. Because I'm going to ask you for answers each table, right? Okay, we'll start over at Pekka's table. So what's your answer?
500 meters.
Okay, 500 meters there. What's the answer here?
500 meters too.
500 meters too, okay, good. And down here? 750 meters, that table? Okay. Let's have a guess. 200 is this table? I'll have a go.
Yeah, 500.
500 meters, okay. So you have 500, 500, 750, and so on. So there's many factors in this answer, right? So let me try and explain a few of the examples. So I might go to the door and I call the cats, and the shy cat just may not come, so it's 0 for her, right? So that happened to me on Sunday. So for Wichigans it was 0. Pixel came though. I eventually found her and we headed off. If it's night, for the shy cat, I have a much better chance of getting further than the day. Or if I go out the time of the day when the neighbor walks the dog, that's the end of the shy cat's walk. And it depends upon many other things, how hot it is and how patient I am and so on. So it varies, and it varies for both cats, and it varies for different times and different days. It varies about what event happens in the pathway above me affects what happens on my pathway with Pixel. There's another factor. Cats are territorial. So none of you actually took that into account, I think, very well. So for a domestic cat, it's about 150 meters. So there's a tree about 150 meters from our house. So even with my non-shy cat, that we reach, and that's the end of the walk. We sort of roar at the next territory, and she goes up the tree, and then we go back. So we never go beyond 150 meters. Right? So the answer is, it's not a normal distribution, right? And there's a lot of uncertainty. And to explain the answer on the uncertainty, we need to understand lots of different ways that each story unfolds, and then we have some kind of answer across all those stories. So when we do risk assessment, this is my attempt, guys, and I might fail. When we do risk assessment against an AOP, it's very similar. We have a set of stories which may have very different answers and outcomes and variations. So the uncertainty is not just some kind of precision or accuracy thing. It may be a different story. And we need to, when we're looking at risks, we need to take all those stories into account, and then both qualitatively and quantitatively against the different events that happen on the story. So my analogy is the walk with the cats. With the risk assessment, it's the chemical in the body or in the environment. We end up at an outcome, but it's a range of many outcomes. So what we have to try and do, and I'm going to do a little experiment this spring with my cats. I'm going to record video, so I have the story part, and I'm also going to collect data. So each walk, I'm going to measure, and record in my diary where each cat got. So I'm going to actually have a much better talk when I try this the second time, maybe, if you give me 3 months. So I'll have the data, and I'll have this distribution, and I'll also have the stories, with the cats. And so we need to, if we're going to communicate to regulators and convince them we have the evidence to make a judgment, we need both the stories and the data, and we need to quantify the uncertainty. So this is a picture of Pekka and me actually taking a break from our meeting today after lunch, and we headed out onto the ferry on the Rhine. Just the two of us, right? And we had a nice few minutes, so we just had a little chat over lunch. It's the reason, actually, I called my blog The Ferryman. And I just want to make the point that in the end, this is a knowledge-based problem, and it's very important that we sit together and take some time to discuss, the complexity of the problems and understand each other, and then come to a better judgment. So that's it. Again, it's a knowledge-based problem. And then I'll just come back to the AOP. So this is kind of like, is this steatosis? Yeah, the allotrope. So actually, there's a lot of work now trying to go on to come up, using all the data we have, with what the AOPs are for different kinds of adverse outcomes. So for example, in chemical safety assessment, we're working on steatosis, fibrosis, phospholipidosis, and we're doing both read-across and ab initio-based risk assessment case studies on these AOPs. So that's already work in progress, just be aware of that. And it's very interesting, but there's a lot of work to do. We may not be sure about whether we have all the events. We need to do text mining to fill in missing events. We need to do kinetics modeling to try and get the extrapolation on a particular event, and so on. So there's a lot of research to do. It's not ready for acceptance yet, but it's on the pathway to acceptance. And I think in the nano safety, and I'm going to pass over to Vladimir in a minute or two, modeling is going to be really important for us to make this knowledge-based risk assessment work. It's not just putting data into a system and building a QSAR, like people thought 10 years ago. We're going to need the modeling to understand what's happening, and to both from a conceptual point of view, and then also using modeling, interacting with experiment to build up our quantitative representation of what's happening in the effects in this system. So with that, I don't know if I succeeded or failed, but I tried. Important thing is we're trying, right? I hope I conveyed, some views about what we need to do knowledge-based nano safety. And now I'm going to hand over to Vladimir, and he'll tell us about all the exciting stuff going on in modeling that I think is going to be a big part of this way to do risk assessment in the future. So Vladimir, please come.